great pleasure to be here today, really. So some of you attended the workshop yesterday. Can you raise your hands for those that attended the workshop yesterday? Some of you attended the workshop yesterday, so can you raise your hands just to know? Okay, thanks. So I'm going to be talking to you about applications of the CRISPR-Cas9 technology and a yeast that is called Candida albicans. So it's a brief introduction. Yeah, I think I'm missing the, yeah, good. As you can see here, it's a polymorphic yeast. That means it undergoes many morphological transitions. Okay, it has morphological plasticity. It can exist in a, a yeast form, spheric, that you see here, but also a filamentous form called hyphae. And this form is important for its ability to cause disease. It is part of the intestinal microbiome, so it's part of the flora. And it's an opportunistic yeast, meaning that it causes infections in immunocompromised patients. And these infections can be superficial, but also systemic. Among the, I don't know why the slides were shifted a bit. So among the superficial infections, you have oral thrush, oral candidiasis, where you see, patches, white patches in the cavity of a patient. But also onychomycosis. These are infections of Candida albicans that are placed around the nail here. Then when in immunocompromised patients, Candida albicans can reach the bloodstream, okay, and cause invasive infections. The presence of Candida in the bloodstream is called candidemia, candidemia, meaning that Candida is present in the bloodstream. And you can see that these hyphal, the hyphal form here, you can see here, is surrounded by, immune cells in the bloodstream. And in the bloodstream, Candida can reach organs, including the eyes, for instance, to cause ocular candidiasis. This is seen in diabetic patients, but also in severely immunocompromised patients. So Candida has a diploid genome consisting of 8 chromosome pairs and around 6,200 pores for a total size of 15.5 megabases. It has no meiosis. There is no meiosis in Candida albicans. And to go from a DNA content of 4N to 2N, you do a concerted loss of chromosomes. There's a concerted and random loss of chromosomes from 4 to 2, okay? So this is called the parasexual cycle. It is characterized by an unstable genome with many instances of loss of heterozygosity and aneuploidy events. But still, the molecular genetics tools are available, including the CRISPR-Cas9 technology. And we're currently constructing, the Candida community is constructing genetic resources for performing functional analysis of genes in Candida albicans. The treatment of Candida infection relies only on 3 classes of systemic antifungal agents. And this is based on the fact that Candida is a eukaryotic species like us. So it's difficult to find a molecule that targets a eukaryotic organism without targeting ourselves. So this is a challenge, basically, to find really new drugs against Candida. 3 families, polyenes that bind ergosterol, disrupt fungal membranes. You have, for instance, amphotericin B as an example. Echinocandins that bind to beta-1,3 glucan synthase to disrupt the fungal cell wall as well. And an example is caspofungin and azoles that bind to 14-alpha inositol demethylase, which inhibit the growth. So this is a biosynthetic pathway of ergosterol. So ergosterol is the cholesterol for yeast species and leishmania as well. And azoles, you have, for example, fluconazole, are fungistatic, and then can see some instances of drug resistance, azole resistance occurring in Candida albicans. So there's, in case of treatment can fail, basically because those drugs can be toxic to patients, and drug resistance may arise, basically. So there are a few options when treatment fails. So there's a need to really develop new antifungal agents. And to that, we need to better understand the molecular determinants of Candida albicans' ability to act as a commensal and as a pathogenic organism. So the sequencing of the Candida albicans genome was performed in 2004, nearly 8 years following the release of the Saccharomyces cerevisiae genome sequence, seminal publication in Science by André Goffaux and collaborating in 1996, with the title "Life with 6,000 Genes," meaning that the first eukaryotic organism to be sequenced is Saccharomyces cerevisiae. This is a budding yeast or bakery yeast, la levure du boulanger. And so it's followed by sequencing of the whole genome of Candida albicans. It's a diploid genome. And the sequence the strain that was sequenced is a strain called SC5314, which is a clinical isolate that is the parent strain of many lab strains currently used, by researchers in the Candida community. So this reference strain that has been sequenced is prototrophic, meaning that it has no specific nutritional requirements. So this hampers, the use of Candida albicans as a species for doing molecular genetics. So we need to create, markers to be able to select for mutants later on. And the question here is how we can use Candida albicans for mutant creation and studying the function of genes in Candida albicans. We need to study the function of genes involved in virulence, in fitness, and its ability to cause disease, but also to propagate in the niches of the human body. So one of the first genetic toolkits for mutant creation were developed by Fungi and Irwin in 1993. And it relies on a tool that was used in Saccharomyces cerevisiae, basically called the URA-Blaster. And the URA-Blaster uses URA3 nutritional marker for selection of mutants, okay? And URA3 allows the growth of Candida albicans on a medium that lacks uridine. Uridine is a sugar version of uracil, which is a component of RNA, of course. And this requires the availability of a strain that is homozygous for URA3 mutation. So we need to remove URA3 to create a strain from SC5314 to be able to do genetics and molecular genetics to create mutants. So the first strain that has been developed by the Candida community and basically fungi is a Chi4 strain. This is a derivative of the SC5314 strain, and we call it auxotrophic, meaning that it cannot use uridine as a source of as a nutritional source. So when you remove uridine from the medium, this mutant, this strain, lab strain, cannot grow. And that's how we can use this marker, URA3 marker, to select for mutants later on, right? Do you understand what is the purpose of that? So the first markers for doing molecular genetics and selection of mutants in albicans is URA3 marker. And how does the system work? So you can see here that we PCR amplify cassette containing the URA3 marker. There are 2 copies of a target gene that we would like to knock out, to remove from the strain. And you flank the URA3 marker by sequences that are homologous to the target. You transform the Chi4 strain, and there is a homologous recombination that occurs. So you integrate the URA3 marker, and then an intramolecular recombination occurs between the direct repeats of the His-G bacterial. These are bacterial components. These are direct repeats. So there is a intermolecular recombination. So the cassette goes and replaces 1 copy of the gene, then intramolecular recombination to loop out the marker, excision of the marker, and then you can use the marker again to delete the 2nd copy. So it's a tedious process, really. So you have to select first, excise, and then reselect again. And we use counter selection by 5-FOA, 5-fluorotic acid. 5-FOA is a toxic compound when URA3 is present. So how to select the mutants that loop out URA3 is by exposing the cells to 5-FOA. It's called counter selection instead of selection. And then, there was development of strains that carry additional nutritional markers, LU2, ARC4, HIS1, and this strain, BWP17, carries a triple exotrophic strains carrying URA3, ARC4, and HIS1. And this allows to use 2 different markers in 1 shot to get homozygous knockouts. Okay, instead of URA3 looping out, transformation again, selection, and looping out, you just use this strain and you introduce HIS1 and LU2. You can use LU2 and HIS1, and in 1 shot, you get homozygous knockout. And then you can verify your mutant by PCR by checking the left side, the right side for HIS1, and checking left side, right side for LU2. You're not supposed to amplify the targeted gene. So this is the classical way to create, mutants using nutritional markers in yeast. Then later on, there was development of dominant selection markers. We're not talking anymore about nutritional markers here, talking about dominant selection markers. And these include the markers for selecting mutants on a drug-containing medium, like nurseotricin, for example, or hygromycin B. So this is nice because you do not need, in this case, strains or create strains that are oxotrophic for certain nutrient components. In this case, you can create mutations and mutants in clinical isolates in prototrophic strains like SC5314 itself. Okay, so you can use a SAT1 or a NAT1 dominant selection markers, and they confer resistance to nurseotricin drug. So it's a marker. It's a nurseotricin acetyltransferase from Streptomyces nursei that is codon optimized and of course can be used for selecting mutants in Candida albicans. So it's tedious to create mutants, homozygous mutants in Candida albicans, because it's a diploid yeast, has 2 allele need to be deleted to get homozygous mutants. There's a need for marker recycling in case of use of nutritional markers like URA3. So there's counter selection, as I was saying, with 5-FOA. In the absence of marker recycling, there's a need for 2 different selection markers. So, for instance, ORG, HIS, or hygromycin and nurseotricin resistance marker. And the efficiency of integration at the targeted locus is relatively low. The success rate is low, basically. So you need to screen many mutants to make sure that the cassette was integrated at the right location. So in 2015, Jerry Fink from Whitehead Institute developed the CRISPR-Cas9 system for genome editing in Candida albicans. And this has basically revolutionized the way we create mutations and mutants in Candida albicans. So Jerry Fink has basically created the system that confers two main advantages. First one is that we can generate homozygous mutations of target gene in a single transformation step, one shot, using the principle that CRISPR-Cas9 allows to create double-strand breaks, site-specific double-strand breaks, and you can release ends that are highly recombinogenic, donc hautement recombinatoire. Okay, so you can insert your cassette and select mutants this way. The second is that the efficiency of integration at the targeted locus is significantly increased by using the CRISPR-Cas9 system. So here is the toolkit, CRISPR-Cas9 toolkit for Candida albicans genome editing. It's one plasmid that carries all of the required elements for genome editing with a stable integration at the N01 locus. So you basically integrate this plasmid that carries the Cas9 codon optimized Cas9. Why I'm saying codon optimized? Because Candida albicans, like other Candida species, has a codon usage that consists of translating the CUG codon into a serine instead of leucine. Serine plus the leucine. And so when it reads CTG, CUG on RNA, you get serine. So if you provide Cas9 from yeast or bacteria, you basically translate into serine and you create a mutant that does not function. So you have to change your codon to allow Candida albicans to translate it into a leucine and not introduce mutations like that. So it's a codon optimized Cas9 version. It's placed on a constitutive and a strong promoter. I think it's N01 promoter. Am I right? Or Act 1, can't remember. Anyway, but so a strong promoter. And you have your guide RNA that you can introduce there. Guide RNA allows Cas9 to, as a guide for Cas9 and allow to induce double-strand breaks. And so in that case, you introduce a repair template, and then you substitute the targeted gene by selection marker. So a selection marker is also available. So this is a NAT1 selection marker that I described previously in a previous slide. And this is integrated, and you could create your strain and perform genome editing in that strain, particular strain. So N01, yeah, this is an N01 promoter, NAT1 marker, and guide RNA that is under the control of this NR52 promoter. This is a RNA polymerase 3 promoter, followed by N01 terminator of transcription. So there's a limitation here, because when you get a strain that carries a continuous CRISPR activity, you could, this could lead to or favor off-target effects. So basically, the nuclease will cut, outside of the region that you targeted. So this is a drawback, really, limitation of the system by integrating the plasmid and the genome. So Aaron Mitchell from University of Georgia has created the transient CRISPR-Cas9 system to streamline basically CRISPR usage in Candida. So it was developed by Aaron Mitchell, and in order to express the system without genome integration. So in other words, you basically use the plasmid as a module to amplify cassettes. Okay, 3 different cassettes are PCR amplified. The first one is a Cas9 cassette with the red and green primers. The second is the selection marker, your repair template that carries 80 base pairs of sequences that are homologous to the targeted gene. So by homologous recombination, you would introduce your marker and get a knockout of your favorite gene. And then the sgRNA with the blue and pink primers, you would perform a fusion PCR by designing your 20 base pair guide RNA and the primer that you'll be using to first amplify the promoter of SNR52. So this is a tail that does not hybridize to any sequence of the plasmid. But this primer here and 1 and 2 would amplify the promoter. You get a tail that carries the 20 base pair sequence that is homologous. So this is the reverse complement of the tail of primer number 3 that would amplify the remaining part of the guide RNA cassette. Then you extend from primer extension and you amplify the whole cassette at the end to get the 20 base pair sequence of your guide RNA, the scaffold RNA, and all of the elements allowing in vivo transcription of your guide RNA. So you basically get 3 modules: Candida optimized Cas9, NAT1 selection marker with primer, so this is template, the prepared template, and the guide RNA under the control of RNA polymerase 3. And you co-transform any clinical isolate that you get, basically virtually. So every strain that you get from patients or a lab strain, you could use that system based on the fact that you're using a dominant selection marker. Right? Okay. Many applications of CRISPR-Cas9, including insertion of genes, to modify the metabolism of a given strain. Deletion of genes thought to be involved in virulence. So this is knocking out a gene in CF. This gene is important for virulence of Candida albicans by performing systemic infection models in mice. So we can inoculate Candida albicans to a mouse, and it mimics the candidemia in patients that are immunocompromised. So there are mice that are immunocompromised, and you could use mice to study the virulence of Candida albicans by knocking out certain determinants of virulence properties of Candida albicans. But also a creation of gene mutations that to confirm the contribution of certain genes to drug resistance. So in Candida albicans, there are many mechanisms of drug resistance, mainly azole resistant, because azoles are fungistatic. So this is the survival. The cells that survive to azoles could randomly get mutations that favor their development in the presence of drug. This is how drug resistance evolves in Candida albicans. But also, this is I will not be talking about this. The CRISPR-A, CRISPR-I. CRISPR-A, CRISPR-I is a great system. I think it's later on we're going to be having a talk to talk to you about the use of Cas9 as like sort of a DNA binding protein, and you can fuse Cas9 to a activation domain. It becomes a transcription factor, an activator of gene expression, or fuse a domain that represses gene expression. So you could, Cas could act as repressor of gene expression as well. And then you can use the system to overexpress or repress gene expression using Cas9 as an artificial regulator of gene expression. Okay, this is a very nice, tool that does not use the nuclease activity, but uses the DNA binding property of Cas9 to modulate gene expression. And this is a timeline of the development of molecular genetics tool in Candida albicans. So in the 70s, of course, we used the mutagenesis through UV, etc., the classical way of forward genetics instead of reverse genetics. And followed by the urablaster, developed by Fonzai and Irwin in 1993. And then there are development of additional nutritional markers. And then you have, I didn't talk to you about that, but to excise a marker or loop out a marker, you could use flip recombinase. So you could catalyze looping out of a marker through enzymatic reaction involved with flip recombinase. It's like the Krelog-Sp system. Have you ever heard of a Krelog-Sp system? So you could remove the marker through Krelog-Sp or flip recombinase. And starting from 2015, we were able to apply CRISPR-Cas9 system in Candida albicans. And then later on with the group of Shapiro in Guelph, I think it's Guelph, Rebecca Shapiro has developed extensively applications of the CRISPR-Cas9 system in Candida albicans. Okay, so quickly here, I'm going to be talking to you about the application of CRISPR-Cas9 editing, genome editing for functional analysis. And I'm going to be talking to you about a project that we developed with Maysa Gnaim, your teacher yesterday during the workshop session. And she's going to be talking to you about this project basically, and she will be performing some training in the lab on how. So you're going to be visualize how we could do that CRISPR-Cas9 to create mutants in Candida albicans. And the project that deals with that is to understand how Candida albicans can basically thrive in a context of cystic fibrosis in the lungs of patients with cystic fibrosis. Candida albicans usually colonizes the oral cavity, the gastrointestinal tract, and the vagina. Okay, but under certain disease context, Candida albicans can increase its colonization rate. For example, inflammatory bowel disease, it increases colonization in the gastrointestinal tract. And in cystic fibrosis, this is a devastating disease caused by mutation in CFTR, cystic fibrosis transmembrane conductance regulator gene, encoding a chloride bicarbonate ion transporter. And this disease is devastating because it allows the colonization by microbial species that cause disease, such as Pseudomonas aeruginosa. And most of those patients die from infections with Pseudomonas aeruginosa. A devastating disease, really. And a young age, they die at a young age. Here in Tunisia, this is really a problem. Because of resources are limited for clinicians. And so we're trying to increase the lifespan of those patients, really. And Candida albicans, so translocates to the lungs. And it chronically colonizes the lungs of those patients. It also contributes to the disease severity. So when we compare the cohort of patients that are chronically colonized by Candida albicans to those that are not colonized by Candida albicans, you see that the severity of the disease is increased in those patients colonized by Candida albicans. And this has been reported by many studies in the past. So the hypothesis of the research is that Candida albicans would undergo specific adaptation mechanisms that explain its chronic growth in the lungs of those patients. And the objective of the study, the project, the MISA's project, is to identify the genetic and phenotypic determinants of Candida albicans' ability to colonize the areas of those patients. So we established a collaboration with clinicians from the Children's Hospital in Tunis, Bashir Hamza. And we recruited 25 cystic fibrosis patients, mainly are pediatric patients. These are antifungal naive patients. They've never been exposed to antifungal drugs. Why I'm saying that? Because antifungal exposure could select for genome rearrangements, aneuploidies, okay? So here there's no exposure to antifungal agents, and we've collaborated with the group of Kalthum Qalel, okay, Parasito Miko Ferrata, and Hanan Smewi from Children's Hospital in Tunis. And we have isolated 146 clinical isolates during 2.5 years. So Mesa was going to hospital carrying the strains. It started in 2016. And we observed Candida albicans colonization in 15 out of 25 patients, which is a carriage of about 60%. We looked at the population structure of these isolates by multi-locus sequence typing. We found that more than 50% of the patients are colonized with strains from clade 4. So clades, like us, people, we are genetically heterogeneous. Candida as well is genetically heterogeneous, and most of the clinical isolates are from clade 4, called clade 4, which is a clade enriched in strains from Middle East Africa. And we found that each patient appears to be colonized with a strain, the same strain, single strain, because they share the same diploid sequence type. Okay, there's no strain mixture of strains. One patient in particular, shown here, was sequentially sampled over a period of 1 year. It carries a clinical isolate from clade 10. So it's a nice model to look at how the strain behaves during time, how it evolves during time in that patient, to try to detect determinants of the ability of Candida albicans to constantly colonize the airways of those patients. So this is a list of whole strains, clinical isolates from that patient, and we extensively phenotype those strains to look for phenotypic changes occurring in that patient that could see, okay, so maybe this strain is adapting to that context, okay? And we found that a subset of these isolates display fuzziness, colony fuzziness. See here that there are like sort of filaments surrounding the colony. So Candida appears to be filamenting. The subset of the isolates from that patient appear to be filamenting. That's the case. You see cellular filamentation is observed in strain HBJ61, strain HBJ63, but not strain HBJ62. So we look at the ability to form biofilms. So some cystic fibrosis associated pathogens, grow in a form of biofilm. This is sticky cells that stick to each other and express a extracellular matrix. This is a form of microbial community that are highly adherent between each other, and they are resistant to antifungal agents. So it's like a sort of a tissue that is constituted by microbial species, a form of resistance to the environment insults. And we found that the Filamentous strain have increased ability to form biofilm as well. And biofilm is a threat that is highly correlated with the ability of albicans to form hyphae as well. So hyphal forms, so filamentous form, and increased ability to form biofilms. And biofilms can be formed between albicans and other species, could be bacterial species as well, okay, in a cystic fibrosis environment. Because cystic fibrosis is highly inflammatory environment, but also it carries many species, microbial species, opportunistic species that could, modulate the severity of the disease, including Pseudomonas aeruginosa, right? So what is the genetic determinant of this filamentous phenotype and the ability to form biofilm compared to the remaining isolates? We sequence the whole set of clinical isolates at the Biomics facility in partnership with Imenajar and Laurent, and by really collaborating with a talented bioinformatician, Corinne Moffret from Institut Pasteur Paris, from the Fungal Biology and Pathogenicity Unit. And you have a map, density of heterozygous SNPs with a 1 kb sliding window that shows indeed that all of the strains carried by these patients are highly related to each other, genetically highly related to each other. So still confirming again that this patient carries isogenic isolates. So the same strain basically that is propagating in that patient. So we've mined those data by comparing, looking at the differences that are specific to the filamentous and strong biofilm formers compared to the non-filamentous isolates in that particular patient. We found 35 non-synonymous heterozygous SNPs and 19 ORFs differentiating the filamentous strain from the remaining isolates. Among these, 13 found in only 7 ORFs, 7 genes, are not present in a set of 182 clinical isolates from non-cystic fibrosis patients, sequenced by Ropars et collègues. This is a group of Christophe D'Enfer, and these isolates are from a variety of sites, the human body, oral cavity, bloodstream, vagina. So these SNPs appear to be associated with cystic fibrosis or correlating with cystic fibrosis context. So we're, narrowing down the list of potential genes that confer this new phenotype in those isolates. And this is a list of those genes. And 2 are interesting for us, basically. 2 genes encoding 2 transcription factors of the zinc cluster family. These are fungal-specific family of regulators that can say that they are like sort of nuclear receptors of fungi, mammalian nuclear receptors, RXR, RAR nuclear receptors. Like zinc cluster act in a similar fashion than nuclear receptors. And then they can sort of acquire mutations that modulate their activity. They can become hyperactive, the gain of function mutation. So our hypothesis is that those 2 transcription factors may carry SNPs or mutations that convert them into a hyperactive state, constitutively active, and this allows to induce filamentous growth in a constitutive fashion and make biofilm. Okay, so we tested this hypothesis. We introduced the mutation. In strain HBJ62, the non-filamentous, non-biofilm former strain, isogenic to 6,3,4,3. And so we found that it induces hyphal, this mutation induces hyphal development, induces the ability to form biofilms stronger biofilms, in the resultant mutant. You could see it in the quantitative assay, but also on qualitative assay. So our hypothesis is that this is a gain of function mutation that in transcription factor called ROB1. Confers constitutive filamentous growth, increased biofilm formation, and our hypothesis also is that it resists to the filamentation repression effect of Pseudomonas aeruginosa. This is a previous work that has been performed by the group of Leah Cowan in Toronto, where she showed that loss of function of a repressor, in this case, transcription factor repressor, could activate constitutive hyphal development and allows Candida albicans to resist to the growth inhibition by Pseudomonas, because we are in the context of competition with Pseudomonas aeruginosa. So in that case, Candida would thrive and resist the repressing effect of Pseudomonas aeruginosa. And this is how Candida albicans is able to chronically stay there. There's no more inhibition by Pseudomonas, and it can grow chronically. So we tested that. We looked at the ability of those strains, HBJ62 plus the ROB1 without mutation, plus ROB1 with the mutation using a dominant selection marker, luciothricin. SC5314 is repressed by PAN1 strain, whereas 6.2 with the mutation in ROB1 is not inhibited anymore. By combining ROB1 mutation and ZFU3 mutation, the other transcription factor that we find as mutated, we see it's even less inhibited by Pseudomonas. So this basically gives a genetic determinant of the ability of Candida albicans to chronically thrive in the context of cystic fibrosis. So now, what is the additional question that we ask? What if we knock out ROB1 from the strain that is hyperfilamentous, hyperbiofilm former, and the strain that resists to Pseudomonas? So we hypothesize that deletion of ROB1 would sensitize 6.3 to Pseudomonas repressible effect, and 6.3 will not form biofilms anymore. So it would phenocopy the strain 6.2 basically. And so Maisa will show you during the afternoon how we design the CRISPR in order to delete ROB1 transcription factor. And so we will be seeing together at the end what is the phenotype that is conferred by deletion of ROB1. So we would see that the strain will not be able to filament anymore, will not be able to form biofilms, and will be basically repressed by Pseudomonas aeruginosa. So our working model here is that Candida albicans can resist to Pseudomonas aeruginosa in the context of cystic fibrosis by selecting mutations in NRG1, loss of function mutations in NRG1 transcription factor, but also by gain of function and transcription factor ROB1 or ZFU3 to resist to growth inhibition and persist in a context of cystic fibrosis. So the future work for us is to really map the transcriptional circuitry of ROB1 using combination of ChIP-C and RNA-seq, look at the dynamics of gene modulation, but by those mutations, mutated transcription factors, identify pathways and mechanisms that underlie the resistance to Pseudomonas aeruginosa. So it maybe this resistance occurs through resistance of quorum sensing molecules released by Pseudomonas aeruginosa, and then testing really the strains carrying those mutations in animal models of cystic fibrosis or organ-on-a-chip models of cystic fibrosis to better understand the mechanistic aspects pertaining to this ability of those mutants or adapted strains, CF adapted strain, in vivo, basically. So that's it. I would like to thank you very much. I'm sorry for getting more time for presenting the project, and I would like to thank all of the team members of my group here at Institut Pasteur Tunis, especially really Mesa, who has did fantastic work since 2016 and coming up with these great data and great results. So Mesa will teach you or like show you how we would generate CRISPR-Cas mutants by performing a deletion of a ROB1 transcription factor in the lab this afternoon. Institut Pasteur in Paris. So I'm affiliated to the group of Christophe d'Enfer in Paris. So there's a really contribution of Institut Pasteur and the fungal biology and pathogenicity group. Of course, my collaborators at Tunis Hospital in Tunis, Kartoum Khalel, Khadija Boussetta, Hanan Smewi, and many members and the patients really who accepted to provide their samples and allow us to investigate how they can carry this continuous and chronic colonization by Candida albicans. Dr. Rebecca Hall for sending a nice protocol that tests the interaction of Candida albicans with Pseudomonas aeruginosa, and funding. So PCI, Programme Collaboratif Interne, Institut Pasteur de Tunis, the fellowship from the Pasteur Network. I'm a Pasteur Network fellow, and the Find Access project that many of you maybe have heard of it. It's a great, really project that allowed us to equip our institution with abilities and skills in analyzing big data and performing functional genomics, and also with equipment, a server that allows us to perform calculations and computational analysis, and also a sequencer that allows NGS sequencer that we equipped recently. And thank you for your attention.
And actually, before the question, that one general question. So we saw here a fantastic and a lot of horizon for therapeutics, but I have a kind of naive question. Is there any opportunity for diagnosis?
For what?
Diagnostic.
Diagnostics. I think we're going to be seeing a talk from Haj Ali about a method that uses CRISPR-12. Am I correct? Hi. So you're going to be presenting some aspects pertaining. Okay. Yeah. But there are applications, but I think Thoraya would also provide a talk that deals with diagnostics or maybe more therapy. But here we're answering two fundamental questions on how microbial pathogens could adapt to certain niches of the body. But basically, what we show here is that zinc cluster transcription factors, since they are specific to fungal species, we could target those factors with compounds. If you inhibit zinc cluster transcription factors, inhibit their function, you would, basically disrupt antifungal resistance, because this is a family of regulators that involved in antifungal resistance. They, in a similar manner that ROB1 and ZFU3, they acquire mutations that convert them into hyperactivators. And this is a similar way and the ability of Candida albicans to develop resistance antifungal agents by selecting mutations, gain of function mutations, and this family of regulators as well. In azole resistance, there's a development of mutations, gain of function mutations, and TAC1 transcription factor, MR1 transcription factor, UPC2 transcription factors. All of them regulate efflux pumps that expulse the azoles outside the cell. So it's a mechanism of resistance. So basically, zinc cluster transcription factors are really like elements that acquire mutations to allow Candida albicans to adapt to conditions that do not favor their growth.
What strikes me first in your exceptional speech, congratulations by the way,
Thank you.
is first the structural similarity in the molecular resistance mechanism between yeast and bacteria. They both rely on biofilm forming capacity.
Mhm.
Second, Candida albicans is a pathogen for fish too, and could give the same clinical feature we have just mentioned before. We could use fish for hypothesis testing and ultimately deploy a One Health strategy for mitigation system.
Yeah, thank you. Really, it's I think biofilm formation is one of the characteristics of microbial species, to resist to conditions that are not favoring their development. So they make like sort of a tissues with the extracellular matrix. The problem is that they are highly resistant to antimicrobial agents. So if we try to find drugs, and in One Health aspects of it, this is biofilms are formed in everywhere in the environment, and even the water supply systems have filled with biofilms. So to find a drug that could target the cells that are encased in the matrix, so they go through the matrix. It's not easy to find a drug that could diffuse within the matrix and reach the cells and to disrupt the pipe. This is a challenge, really. But by inhibiting the strategy of using zinc cluster transcription factors as a target for antifungal development is really attractive. It's kinases could be a good way as well, because the kinases are good nice drug targets for pharmacologists, because they have the structure in pocket that allow to be druggable. Transcription factors are more tricky to use as targets for development of drugs, but why not? Well, I think clinical model for Candida albicans, the future is on an organ on a chip. Those organoids. I think one could develop a long on a chip system where you could try to see if Candida albicans would form biofilms with Pseudomonas. So this is a nice, we're not showing here, but maybe the thing is that Candida albicans is able with those gain of function mutation, not only to resist to Pseudomonas inhibition, but also form biofilms with Pseudomonas aeruginosa in the context of cystic fibrosis. So this is how it stays chronic there. Yeah.
Thank you for the very thoughts. So my question, do you intend to do post-pathogen interaction to see why some patients are expressing or have favored therapy for some candidate brain? For example, patients are presenting Candida strain 4 and patient presenting strain 10. This is my first question. The second question is about confirming that these strains are associated to cystic fibrosis while the other strains are presenting other patient numbers for other population. We intend to go through and do an epidemiological study and see patient and healthy controls from the same environment and the same population to compare whether these strains are actually strains specific for cystic fibrosis in Tunisian patients.
Yeah, thank you so much. Nice questions. So the first question is, you said quickly, host pathogen interactions. Yeah, so testing host pathogen interactions. How you could test that? Basically, by developing models of cystic fibrosis and an animal model of cystic fibrosis to look at the interaction between the host and the pathogen. You could not do that in humans, right? For ethical considerations. But here we're collecting clinical isolates from patients. So it's at least a relevant model to look at the evolution of the strains in the context of host pathogen interactions. And I think the best model to use for now is not mouse or other rat, or because they do not develop cystic fibrosis symptoms like humans. It's a bit different. They have CFTR. We knock that. You can knock out CFTR, but mice do not develop exactly the same symptoms. So it's not really relevant. A system that you could really use to look at the host and the pathogen interaction is, as I was saying, for long on a chair. Long on a chair, you have exactly the same composition with vessels, with tissues, with everything related to the lungs, and a microscope slide, really. And it's connected with this complex setting that allows you to mimic exactly the situation of and the function, the physiological or pathological functioning of the lung. In the cystic fibrosis context, you could take CFTR cells and grow them, and then deposit Candida albicans, Pseudomonas, and add additional microbial species. And then you could look at the host and a pathogen. Look at cytokines, markers, look at the response of the lung to additional Candida, what happens, additional Pseudomonas, what happens, both, what happens. So you could play on the different parameters, look at the interaction of microbial species with the host. Your second question, you said, I don't know if you answered the question, first question.
Yes.
Yeah.
But what about the genome of the host and
Genome of the host, well, you already have the CFTR mutation, and so it's very complex. So, for instance, you said that. So, second question is related to second question. You have strains that belong to other clades, mainly four. So, and if there, you said, question is if there's a transmission between patients. Candida albicans, we got it right away after birth from our mother. Okay, usually our mother could provide her strain, the strain that colonizes the vagina, could be transmitted to the child. And we looked at that aspect with mice. I think we looked at strains from their mother as well to see if there's a transmission from the mother to child. And if this is the case, would be an outstanding control strain. Control strain is a strain from the mother, that isogenic strain that is transmitted to the patient. And then you would look at the evolution of the strain in the CF patient and use the same strain that is from mother that colonizes the vagina. So you have the same strain colonizing healthy individual, and it's the same strain colonizing the see application and look at that. But it's more complicated than that. We have different phenotypes in other set of strains. For instance, clade 4, we found a different phenotype. It's related to cell wall. So we're going to be investigating more of that. Well, so this, heterogeneity of adaptation mechanisms as well as expected.
When? So, thank you very much.